Just going to bring up my screen. Yeah, do you want to join? Yeah. To the side. Okay. So, can everyone see my screen? Okay. Fantastic. Well, just to quickly run through the agenda, I'm going to give a brief introduction and an update on where things are at with Boffin. And then we'll run through the results, starting with the soil core samples with Craig, moving on to the microbial analysis with Maria, and then looking at the AMF samples with Tom. And then hopefully there'll be time at the end for any questions. Just to let you know, in case I forget at the end, the reports will be going out to you sometime after the webinar today. So if you've got any further questions once your reports come through to you, then please do put them on the soil circle, and we'll make sure that you get an answer. So just quickly, just to run through, you probably have noticed that there's new payment rates for this year. So this is something that we've successfully negotiated with Innovate, that there's a rise in the day rate, which affects the amount that you get paid. The effect is that you get an extra £1,000, which is great news. Now, it does have to be said, it does result in an extra membership fee, and the membership fee is £750. So that reflects the extra costs that we do in all the marketing and promotion, dissemination activity, and so on. We pay a share of those costs, so that's to recover those costs. But that's a net payment, an extra net payment for you of £250. In order to claim that, then I do need an extra invoice from you, please. You've all sent in your first invoice. You've received your first instalment, or should have done, by the 30th of December. You've paid your membership fee, or you should have done. Then your second instalment will come through on the 30th of September at the end of the trial this year, and there'll be an extra membership fee that'll come through from that. So the action, as far as you're concerned, is that if you could send in an invoice any time between now and the end of the trial, and then you will receive your extra payment, and you'll receive your extra membership fee as well. So just quickly also to note that we have started Boffin Growth Partners, which is a community interest company owned by its members. This is going to give more opportunities for you to take part in trials, we hope. And also we want to set it up as an independent research organisation, so to recognise the value of farmer-led research. But there'll be more on that as it progresses this year. And just quickly to flag up some diary dates, and to flag up our inspiration days, which will be taking place in May. So on the 8th of May, there's the East Inspiration Day at Thrale's End in Harpenden in Herts. Then we got the West Inspiration Day, which is taking place on my farm, Whipplesham Farm near Farringdon and Oxon. And then we've got the North Inspiration Day taking place at the Arbiki Distillery near Dundee in Scotland. So that should be a really good day for those people in Scotland. And then also to flag up the cereals event, we're going to have a stand at cereals. Really want as many of you to join us as possible. That there should be a few announcements and things. We're hoping that we're going to be involved in quite a few cereals activities as well. So that's shaping up to be a really interesting day. We're just getting the resource packs ready for sending out shortly for this current year, just updating all the resources. So those will be coming out to you within the next month, so that you can get underway with your trials. Just to give you an update on my trial, I don't know whether it was a good idea, to be honest. We've grazed it with sheep in possibly the wettest week of the whole year. So I panicked after they'd been on there for about 2 or 3 days and took them off. So hopefully no long-term damage. This is what it looked like on the 5th of February. You can just see the the Watkins 238 in the foreground next to the Graham, which is the farm standard wheat. And then you can see there are still some plants out there. So I think it's survived the sheep. So it'll be interesting to see how the grazed fares against the ungrazed. Bit of a new departure. But I know that there are plenty of people on the call who do this routinely. It's a bit of a new thing to me, and I'm really worried that it's not going to come back. But let's see what happens. Anyway, that's enough from me. I'm going to pass over to the real experts on the call here, and I think we're starting with. Hang on, I'll stop sharing my screen, and we're starting with you. I think, Craig.
Yep. Thanks, Tom. I'll just... That'll come through. Yeah. Yeah, I'll take it. That's all okay.
Yeah, we can see Craig.
Yeah, that's good. Okay, morning everyone. So this is a few slides really on the X-ray CT images of the soil cores that you sent in. This is kind of put together by the team at Nottingham. I'm Craig Sturrock, and there's also, we've got Sasha Mooney and Brian Atkinson that's also part of the team here. Sorry, there we go. So just to remind any of the new route rangers, I guess a few of you have probably seen some of these slides before from last year that took part in the project, but we're using this imaging technique called X-ray computed tomography, and it's very much like a kind of medical CT type instrument where we're collecting non-destructive images of these soil cores that you've sent in. And I mean, the technique's great because you can actually see inside the soil cores and see where all the soil pores are and see where the plant roots are. So yeah, this is our system here. We kind of pop the soil core in, we expose it to X-rays, collect a whole load of images. But the key thing is here, we get these sort of cross-section images of what the soil cores look like inside. So you can, it's quite a nice example here. There's a cross-section through an earthworm. We've got the outer edge of the core. So this kind of cutting it in half basically and looking at what's inside. But what we're really interested in is where all these darker regions are, the soil pores, and the roots are tucked away inside there as well. And we can really kind of visualize these in 3 dimensions. So there's lots of ability to really kind of delve into that structure. It's a bit slow. There you go. So, I mean, I think all these images are going to be shared, so you've got time to kind of delve into them a little bit more detail. But say we're kind of classifying what different materials are in these X-ray images, so we can kind of get where the pores are. The higher density stones are always the brightest. You can see where the roots are, and maybe earthworms and things like that inside it as well. This year we've been using this new kind of analysis technique that was slightly different than last year, and it's really kind of given us some additional information on things like how connected the pores are and impacting on things like water permeability through the soil. So I'm not, it's quite a lot of detail in this. I'm not going to spend too much time because I've got some examples here. So I've just picked up one of the cores that had a really kind of nice pore structure from Rachel's farm, Rachel Holmes. This is core number 4. And I mean, this example here, we've got all these nice open pores here, and there's some heavier kind of higher density objects in here as well. And you can see the kind of roots crown at the top, sort of cross section through the roots. So this is just one cut through this soil core, and you can kind of get an example here of what that looks like in 3D. So when we're doing this imaging, first thing we do is we kind of separate out what's solid and what's not. So basically turn it into a black and white image. So again, all the black here is all the pores, all the soil's just now white. We invert it, crop a bit of the background off, and then we can start to measure things. So like the porosity is just the ratio of how much white pixels there are compared to how many dark pixels here. The surface area is just obviously the area that's covered by all the pore surface. So these are going to be really critical for habitat space for microorganisms, how the water and nutrients are going to flow through that soil. Then we're going to kind of a bit more complex ones. There's things like the pore connectivity. So these gives us a measurement of how big this pore network is and how connected it is and how kind of convoluted or complex that structure is. And that's going to have impact on how it functions as well. Some of the other ones, we've got kind of pore thickness. This connection probability is a kind of percentage of how much of pores in this soil are connected together. This one is very connected. So 78% of this porosity is connected together. But interesting, there's a couple of other ones here percolating. It's either it's 0, it isn't percolating, or 1 that it is. And this I'll kind of showcase this in a minute. So thinking about this going from here, a 2D image into 3D, it's kind of got all the soil stones and things you can see there, some of the pores and voids. Here we just pulled out all that pore space. There's a lot in this one, like 10% of that total volume is pores. But interestingly, with this one, although it's really porous, it's not, we don't have one big pore that connects it from the top to the bottom. So all the blue here is kind of that big pore. So this is why it's not percolating in this case. And we can kind of see this maybe in a little bit more detail here. This big pore network kind of stops about just over 75% of the way down. If it's connected from the top to the bottom, this percolating value would be 1. And it would also give us a value here of what the critical pore diameter is. And this is the smallest pore that connects the top to the bottom. So, I mean, if this went all the way down to the bottom, it might be in here somewhere. So it just gives you a bit of background on how to interpret the data. Again, we've got lots of other root rangers this year. We've got a whole range of different soil textures, different cultivation practices here. So we've kind of put all this into the report that will be sent out and bringing some of this stuff together. And now I've kind of got a few examples of some of the images. So I'm just going to use the codes because I can't remember everyone's names and details. So I just hope that's coming up. So AB2, we've got, interestingly with this one, there's quite a good aggregated structure here, but I think these were probably taken when it was quite dry. Obviously the moisture level in the soil was the real problem because of the drought that came in really pretty early on. So I know that it's big challenges in actually getting these soil cores out in one piece. So when they've been cored here, there's obviously a lot of kind of cracks that you can kind of see round about here. That, I mean, the porosity is very high compared to the kind of the mean values across all the root rangers, but where it's much higher, I think it's probably attributed to this sort of fracture that you can kind of see. Irrespective of this, there's still really good kind of rooting. You can kind of see we pulled these out in 3D. Lots and lots of roots coming out in the crown here. They maybe don't look particularly deep, but I think it's the 3D aspect of this that when the roots get cut while you're taking the soil core, they probably go out further and deeper into the soil profile, but we're only really capturing the bits in the sort of an immediate kind of crown region. But I mean, we've got, these are quite kind of wide roots rather than kind of steep ones in this variety. More consolidated soil structure here, but lots of evidence of big biopores here. You can actually see this, like there's something actually, some kind of burrowing insect or something in there. There's an earthworm in this one. It's a quite large biopore. So a good mix of kind of structure in these samples here, kind of maybe deeper roots going down here. So I mean, it's quite interesting to see that variety. I mean, there's some with more plants, maybe some with less plants in them as well. Yeah, quite variable soil structure. Maybe a few more stones in this one. Some of the cores are in good condition when they've come out. Maybe this one's a bit more fractured and it's kind of come loose. You could say that could just be due to the shipping rather than the cores, how they've been sampled or anything. Bit of variability in height here, but it's always a bit difficult. Obviously, we're completely aware of the challenge in getting the cores here in one piece, but so two of these ones, again, it's quite a nice variable soil structure here, lots of kind of individual aggregate structure you can see here, but still lots of biopores. I mean, the root crown here, there's big biopores going through there. I'm kind of conscious of time, there's a lot to kind of go through, and kind of deep variable kind of root structure here, lots of pores. This one much more sort of stonier. So here's some quite large stones. It's quite impressive to actually have gotten these ones out. Obviously, this is impacted on how the roots are been growing. They're probably growing completely around that pore here because they can't go through it. But yeah, some nice root structures here. Again, a bit more of a kind of consolidated structure here, but still lots and lots of biopores, lots of aggregates near the sort of soil surface as well, indicating kind of good structure there. Good rooting depth down through the core. Another one that's maybe a bit more fractured here. Just probably, I think maybe drier soil conditions here. Also, the soil texture also impacts in this as well. But I mean, it's quite stony, quite a challenging one. This one maybe. So it was kind of difficult to actually pull out where the roots were in this. This one was really interesting because it's got almost like a kind of two-phase structure on it. You've kind of got a bit more consolidated structure in the bottom half with kind of nice biopores going through them, but then this really kind of finely kind of aggregated structure on the top, which was really interesting to see. But yeah, I mean, the biopores going right down through there, lots of good rooting there as well, so it's good. Another one, kind of quite a lot of stony soil here, but again, quite a mixture of nice open porosity in the top and then good kind of biopores going through here. There's kind of consistency. If there's all the cores that came in had kind of quite a good high level of biopores through them. Don't know how I'm doing for time. I'm kind of realize it's somebody in Boffin can shout at me when I'm nearing.
Yeah, you've got time, Craig.
Let's go.
I think it's until 10 to, so don't worry.
Yeah. So yeah, this one quite a nice mixture of kind of different rooting depths and things here. Quite a few stones through there. Again, it's similar, kind of slightly different, but more kind of aggregated structure through here. I think it's maybe slightly drier when these ones were taken. So some kind of horizontal cracks going through there. Still reasonable rooting. Again, kind of used one of Rachel Holmes' cores earlier on, this core 4, but I mean the level of biopores through these ones are really quite extensive, so the porosity is quite high, and I mean it's convincing that it's real porosity, not due to any kind of additional cracks that you've got through. So I mean it probably helps with the soil texture as well, but yeah, it's really always really nice to see these ones. The core 3 in particular seems to be completely riddled with pores. Interesting with this one, there was quite really one of the cores had quite thick roots compared to maybe the other ones. Well, this one's quite thick as well, actually. So it was interesting to see. We were kind of trying to see whether or not they were interacting with the biopores, but they're kind of just going through that soil profile. So that was interesting to see. A bit more of a consolidated structure here, maybe some finer cracks, kind of aggregation through that one, but still kind of big biopores there. Here again, quite a heavy clay soil here, more a consolidated structure, but you've still got that kind of aggregates on the top, kind of higher porosity here, although it's kind of more condensed, I guess, going down through here. It doesn't necessarily mean it's compacted, it's a kind of more tighter knit soil structure in the CT images. Dead ones, yeah, they've quite a lot of big chalky stone, flinty stones in these ones, so they're always quite difficult to come out due to the location that they are. Still quite big biopores going through there. Again, big challenges getting these ones out. There are a lot of large stones. I mean, you can see the cores here have fractured. I think these ones were sent in maybe a bit later in the season due to the drought conditions made it was probably really quite difficult to see, but still like really good roots coming down through that. So I mean, the plants are still doing quite well. There's lots of graphs in here that we've just sort of compared how your own measurements relate to the mean across all, and maybe how some of them are quite high amounts of variation. So the error bars here relate to how much difference you've got across the 5 cores that were sent in. Some of them are a bit more consistent, some of them are a bit more variable. I mean, we've just got porosity, surface area, this pore thickness, how connected the networks are, this sort of percolation threshold. So kind of as you go through this, you can kind of see for yourself how your own cores kind of compare with other ones. But I think in general, I mean, the key message is there's really good rooting structure through the years and good kind of aggregated structures as well. We've kind of measured the crown root angle as well, so you can kind of maybe see where there's deeper or shallow rooting varieties. Or so obviously, I think the key thing about last year, it was so dry that it made his impact on how well you could actually get the cores out of the ground. So really thinking about this year, hopefully it's a bit easier. Certainly just now it's a lot still. The soil's going to be nice and moist, but maybe too moist. But we just need to bear in mind that some of the measurements from this year's data, the confidence in whether or not they're true or due to kind of fractures and things are going to be in there. I mean, again, lots of biopores through there, kind of indicate a really sort of healthy soils, improved water, and kind of deeper into the soil profile. And also, like, most of the roots looked really good. And I mean, we've still to sort of pull together these CT images with the Shovelomics data and see what inferences we can draw there. Okay, so I mean, that's quite a quick run through of that, but hopefully it's often been interesting, and I'm happy to answer questions at the end when we have the discussion session.
Thank you very much, Craig, and we'll move straight on to Maria, if you'd like to bring your slides up, Maria.
Absolutely, thank you. And great presentation, Craig. I hope I can link it nicely with my own results. So as you know, well, welcome everybody. Really nice of you to join us this morning, and thank you for all the samples you have been collecting through the season. That has made this work possible, and I hope my quick overview is informative. I have prepared a few introduction slides so we can have a bit of a context in the results we are going to see. Obviously, you are going to receive at some time soon a proper report so you can look in detail, and obviously these slides are available to all of you. Because this year I have taken a bit of a different approach, I have tried to make it also more individual. And obviously there is still plenty of analysis to do in the results. With that said, let's dive in. If I can manage yeah. So this is as a quick overview of your farms of all the different sites. I hope the coordinates are right, and you probably are capable of identifying yourself by the location. So although some of you provide a description of your soil, Agritech has run, I believe it's Agritech or Bofen, Laura, you correct me. They did some analysis with Landcrop, which has also come with a soil texture analysis. So this year I have classified them according to that information. And this is just an overview of your farm. So you have a big number of regenerative, is kind of the most dominant one. And obviously the definition of regenerative is very much broad, and you know better than anyone who is doing that, organic and a few conventional ones. And as you know, our strategy was to sample for the microbiome analysis, for the microbes in the soil. We wanted to sample before and after fertilizer application. For some of you, that was a bit of a challenge. For some of you, there is no fertilizer. And this year, we have focused only on rhizosphere. For those that are returning, you know, last year we were looking at margin and bare soil as a control. So this year, we decided to focus the effort on rhizosphere soil, which is the course you sent me with all the roots and the soil. And well, this is just an overview of all this analysis that have been done, from characterization of the soil chemistry to the CT scans that Craig has been presented, and the mycorrhizal fungi that Tom will present after me. And this first chunk here is me. And this is basically what happens. So you collect your soil sample. It looks a lot nicer here than it probably was doing it in the field itself. Then this is what I do. I just extract all the DNA from any microbe or organism that is in the soil. Then there is a few steps of preparing the samples for sequencing itself, and then the data come back to me and I try to make a bit of sense of it depending on the different parameters we want to look at. So we obviously have an overview of taxonomy, which means which bacteria are there, which fungi are there, and any other organisms of interest. But instead of just giving a table of names, which some of them we know very well what they do or not, we can actually look with a shotgun analysis. We can actually look at what genes all these organisms carry and which potential function they are playing in the soil, and not just the genes, but also which metabolism is happening. And well, obviously all this information can be used for predicting nutrient cycling, if you have a good condition for plant growth promotion, etc. Why am I interested? From all the genes that we can, Simon is here, that's on time for me. From all the different genes that we can pull, there are metabolic pathways for nitrogen transformation, carbon transformation, phosphorus mobilization, a lot of many important things. So my main interest, and I think it's also interesting for you, is to look at nutrient cycling and carbon turnover. But for this project in particular, I wanted to complement the shotgun with nitrification rate. So we just incubate the soil with ammonium sulfate. We measure how much nitrate is produced at which speed. And very briefly, why is this important? There is a lot of controversy that seems like nitrification is evil, but it's actually there are positive things about it because it's a natural process. It's just that if it occurs too quickly, it will result in fertilizer losses and contamination. So very briefly, what we want to reduce a bit, what we would like to see reduced with either land management or nitrification inhibitors or new root traits, will be a slowing down of this process. So things I have taken into account, considering the number of samples that we have from your sites. So obviously you have different soil type. That's going to be the main dominant in which microbiome we are going to have. And that links with what Craig was explaining about microhabitats and so on. The level of organic matter and how that relates to the soil type will be really determining the richness and the abundance of your microbes. Climate, I think we know it's really, really important. Also, obviously, if you remember the map, you kind of go from very north to very south of UK. And of course, the thing that I find more intriguing, which is the land management. So with that, we will have a combination of things, and plus the different wheat that you're going to have is going to release different compounds, and that's something we will be digging a bit more with the 4 varieties we are growing this year. So this is just a nice image of how all these factors and all the inputs from the roots in the soil are going to actually shape which microbes you have and which functions are they having. And then in this, I'm not going to go in detail. I want to show you some of your data, but different combinations of soil and management. For instance, if you have a sandy soil and organic management, your microbes are going to be dispersed. They are going to have less opportunity to find themselves in a nice microhabitat. So they are going to be much more dispersed. So your management is going to have a less impact than in a clay soil where communities can be much closer together and interact. So something to think about when we interpret the data. So basically, clay soil and organic management is a situation when we can actually have much more control of which microbes we are recruiting in the rhizosphere of our wheat, in this case, of other crops. I just dropped this here because obviously, I think we all know this was this is across this year. This is by root ranger and by month through the growing season. So you can see we already have the factor of the different climate across your farms to consider when we compare microbes across the different root ranges. And just for the ones that were recurrent this year that they came back, so I took the minute to compare the weather between 24 and 25. So you can later on, we can compare how it has evolved the microbes in your farm through the years. And obviously here we can see the big drought of last year. This is quite apparent. And for your personal root ranger information, I have selected these few things because obviously we have a limited time. By the way, before I dive into this, Laura, how long do I have?
Until 5 past, I think. Is that right, Sky?
Yeah.
Yeah.
Maybe 6-5 if you can. So we are looking at diversity, which is just all the different microbes they are and how distributed they are. Higher diversity initially means more resilient soil that can can have a resilience with biotic and abiotic stresses. Your fungal bacteria ratio is something that it's also quite important because it will be linked to soil structure, water storage, carbon storage. I did want to play on because actinobacteria and firmicutes are some of the main taxa that you will see in agricultural soils. But I think in this year particularly it's interesting to see which role they were having in your crop resilience to drought. Then, of course, like I was saying, nitrogen cycling genes, how your management, your soil type, and your climate is affecting how much or how quick your nitrogen is being transformed in your soil. Impact of soil type and impact of management, as I have been repeating, is important. So now you will soon have a report with this and other parameters. I just want to, so obviously there is no time to go through each one individually. I'm sure I can come back. There is a slide like this for each one of you that I had enough samples. And so you will find your acronym. For instance, this is Andrew, I believe. Farm number, as I have coded it for future references, your soil type, pH, organic matter, and if you were new or returning. And you will find yourself in the red bar. So for instance, in this case, alphabetically, you get to see your first self first. The diversity we will have, if it's above average, as we can say here, and compared to the rest of the farms, similar to the fungal bacteria ratio. And not just that, but also we have two rates of proteobacteria versus actinobacteria, proteobacteria versus acidobacteria. When we are building the report at the minute, I think we can expand a bit of that, and you can always ask me what that means later on. I'm always looking, every day I look at the soil circle, so if you have a question for me, more than happy. Then you can also see what your drought resilience was, considering the amount of actinobacteria and firmicutes. Particularly the firmicutes, they are quite desiccation tolerant, so they can come back quite easily, and it's kind of a buffer. And I think what is interesting also is then to relate this nitrogen cycling thing. So you will be here in the middle. So I think it's a good balance. So overall, if you look at your soil type, these parameters and your management, which you are in this case are regenerative, basically overall it suggests an effective soil biology management. So I think whatever is happening in this farm is we manage quite well. So we have a more resilient panorama scenario and quite average, almost high ratios for the microbiome diversity and quite a middle nitrification or nitrogen cycling functions. So I think you get the idea. So before I run out of time, I think you can see all of them here. I'm sure you're curious to see your own. But I have a couple of slides that I also wanted to show, and it's just in the nitrification rate, because obviously we have some troubles to measure all of the samples. Some of them there was not enough soil, and some of them was not properly. But basically, I have grouped all your nitrification rates into regenerative, organic, and conventional, and we can see very different like last year. Last year, we have very striking differences. Conventional was showing much more nitrification rates compared to regenerative or organic. Well, this year, although we see some changes like with fertilizer response and the management type, it's not as striking. The only thing that has stayed similar, and that is because this year it was incredibly dry, organisms are very dormant. So we are really trying to deep into the results to see the differences, but we are going to find much more flat differences because the soils were too dry for microbial activity. So we probably miss actually that change with the fertilizer. However, you can see that regenerative and organic pre and post fertilizer from the data we have are not that strikingly different. So there is much less, they're more resilient to the application of whatever fertilization, while conventional, again, like last year, like in 2024, sorry, last year for me is the previous year, we see that the application of the fertilizer causes a real strong response. And that's, again, the same message I was giving in the previous season. The soil is much less resilient. It's going to get conventional practices are resulting in a really quick increase of the nitrogen transformation that is causing a risk of losses of the fertilizer and contamination. And then I have it by soil type, which is obviously we are going to find. So sandy silt loam, is the one that is more susceptible to also this quick transformation of the nitrogen. And with that, I think it's my time. So I'm happy to come back to any of the slides, happy to chat with you. I just wanted to say thank you to all of you that has taken into growing the Watkins and the Duron lines this year. And I very much look forward to the end of the season when we can discuss the results of these lines. Thank you.
Brilliant. Thank you so much, Maria. And sorry that we are rushing you guys. There's so much interesting content there. But yeah, just time. Can I hand over to Tom Thurkill now?
Yep. Thank you very much. Just find my slides. Hopefully you can see those.
Yep, we can see them.
Brilliant. Thank you. Okay, hi everyone. So I'm going to talk through the results from the mycorrhizal fungal colonization analysis that we've been doing here at the Crop Science Centre. And we've had a fair bit of turnover in staff, but I'd like to thank especially Rachel Knox and Sonia Powley have been helping me with these samples. So very quick recap first to say that in most cases, plants don't have roots, they have mycorrhizas. And it's worth remembering, I think, that most plants are mycorrhizal, including all of the top 10 crops worldwide. So they're there. And as a result, arbuscular mycorrhizal fungi are pretty much everywhere in arable soils. So when you plant these crops which are susceptible, including wheat, they will become colonized. That's the sort of default status for them, is to associate with these fungi. So we need to know what they're doing. And we know embarrassingly little still, I would say, for how long we've known about them. And they're incredibly abundant as well. So a kilogram of soil can contain 30 kilometers of the fungal hyphae, these filaments growing through the soil. They're also diverse. We know that there are at least 200 different species of these fungi. And we also know that they're really important in some cases for plant nutrient uptake and defense response. So they can provide a plant with 100% of its phosphorus uptake and make big contribution to plant nitrogen uptake. And they have sort of wider effects on the ecosystem as well. So they've been dubbed ecosystem engineers. They're big players. And we also know that abundant and diverse mycorrhizal networks in soils are probably most beneficial to their plant hosts and to the wider environment. On the other hand, chemical, physical, and biological disturbances can all reduce this abundance and diversity. So what we really need to know is the mycorrhizas which are in arable soils, what are they doing and how beneficial are they? And really, actually, I should have put in here, we need to go back a step even further and have a good characterization of how many of them are there. What's the abundance in these soils, which is where this project comes in. And then ultimately, it would be great if we could sort of work to identify this Goldilocks zone of disturbance where, yes, the soils are being managed for cropping. We also have reduced disturbance where our benefits from the mycorrhizal fungi are maintained. And that means we can hopefully rely less on our bagged fertilizer inputs or our inputs of other kinds. So what did we do with the samples that you sent in? First stage was to re-wet them, so they all arrived in dry sample tubes, and to allow the selective staining processes so that we can see these fungi, we needed to re-wet them, so they were incubated just in some tap water for a bit. And then the nasty chemicals started, so we clear them in potassium hydroxide at 80 degrees, and this dissolves lots of the pectins and the celluloses, hemicelluloses in our plant root tissues. And this allows the staining solutions to sort of permeate and percolate through the roots and get into the fungi. And then we need to acidify the samples, so they then put into hydrochloric acid for a brief amount of time, otherwise they will just disintegrate and disappear. And then we use our staining solution, which is this trypan blue stain, which is really neat because it selectively stains fungal tissue. It actually stains the chitin in the fungal cell walls. So if there are any little insects accidentally find their way into these samples, they will also go bright blue because their exoskeleton is also chitin, but we're using it for the fungal tissue here. So staining with trypan blue and then mounting onto microscope slides. So we'll have 10 or 15 sections of roots. Each of the slides is from one sample tube that you sent us. And then we'll be doing, or we have done, some microscopy and photography to quantify and characterize the mycorrhizal colonization. And I'll just quickly go through each of the structures before presenting the data. The main structure we see, the most common structure we see, are these intraradical hyphae. And hopefully you can see we've got two sections of root here, the boundaries of the root, and then on the inside we have this darker blue staining of fungal hyphae. And this is the first structure that will grow inside the root after the spore in the soil has germinated and made contact with the root. Fungus is growing all through the roots. And this can be really quite prolific. So it allows not only transport of nutrients up and down the root, within the fungus, but also it allows the exchange between the plant and the fungus. And hopefully you can see from these two pictures the sort of contrasting shapes and sizes of some of these fungi. Sometimes they're very thin, thread-like hyphae like this on the left, and other times they're much sort of rounder and fatter tubes, as seen on the right. So we haven't done any DNA sequencing on these, but almost certainly these are two different species of fungi in these root samples. And another thing to say is that the extent of this colonization, which is a sort of theme through this entire talk, is highly variable within and between different root fragments. So what do we see in the samples? So this graph is showing the total colonization, so these hyphae, as a percentage of the root. So this section on the top right, this image just indicative of what we're looking at, this would be scored as 100% because if this whole section of root looked like this, because there are these hyphae through the whole thing. Each of the dots on the graph, each of these circles, represents the sample from one tube. So each farmer, I think you sent in 8 samples each. So we've got 8 sample tubes represented here. And then the boxes really just help show the distribution of that data and see how spread out or how compact it is. And hopefully it's really obvious from the start that we've got a lot of variability between farms and also within. So again, so I don't have committed to memory all of the acronyms, but farm AB2, the lowest sample number was just under 20% of the root colonized, and the highest was up at 80. And I would say, actually, I was really surprised by these samples as a whole sort of class, because what I would expect is that most of these numbers would be about 20%, but actually what's demonstrated here is that there's a lot of mycorrhizal fungi, which is, to me, very exciting to see. Hopefully it's exciting to you too, but very promising, I think, to see such high colonization. In some samples, there were none lower than sort of 40 or 50%, which I think is really incredible. And lots of people in plant sciences and mycorrhizal sciences and the like who would tell you that plants like wheat, because they've got very fibrous and fine root systems, they don't really need mycorrhizal fungi. They don't really engage. Your samples are really clearly demonstrating to us that that's not true. There's lots of fungi there, and that's great to see. So moving, sort of zooming in to the scale of one single plant cell, we're looking for these arbuscules that the fungi create. And these are little tree-shaped structures, and they're very highly branched. So this makes a high surface area, allows the plant and the fungus to exchange those nutrients, and hopefully give us this mutually beneficial symbiosis. And hopefully on this middle image, you can see the cell wall has picked up a bit of the stain, the plant cell wall, but in the middle is this sort of cloud-like, tree-like structure. And eventually this will grow to fill the entire cell. So up the top, you've got 4 cells in a row that are completely filled with fungal tissue. Huge surface area for nutrient exchange there. And because we've put these plants, these roots through these harsh chemical stains, and everything you're seeing is very much dead, we can't tell what's going on currently, but they give us an idea of the potential nutrient exchange. So without these structures, the plant and the fungus can't exchange nutrients. There's no guarantee that they are exchanging, but it's a very, very good clue. And also something to point out that this is a real snapshot. So these arbuscules will last maybe 2 or 3 days each, and then the fungi disintegrate them, move along the root a bit, and then they'll grow some more. So what we're seeing at any time is a snapshot. So if your samples were taken 2 weeks earlier, 2 weeks later, we would expect to see the numbers be quite different on these graphs. The trends might be similar, but I would expect the numbers to be different. So it's just always bearing that in mind. And what do we see in the data? Again, I would say generally higher than I expected. Previous work I've done puts these numbers sort of on average down at 10% maybe, if you're lucky. We've got some really astonishingly high numbers here, I would say, which is suggesting to me that your wheat plants are really exchanging a lot of nutrients with the mycorrhizas, and also relying on them to get their nutrients, which is very exciting. Again, sort of as we go through the presentation, the data get more spread out because these structures become rarer. So you do every so often get much higher numbers, but again, hopefully you can see some of them really incredibly high, which is great to see. And again, lots of variation between samples, between farmers. And then lastly, just quickly talk about the vesicles. These are large lipid stores within the root that the fungus creates, because the fungus can't create some of its own lipids in the way that we need to get some of our vitamins from our food, because we can't create them ourselves. The fungi need to get these lipids from the plant. So it stores them in these huge vesicles, and these are helpful because the supply of carbon from the plant is not constant, for a start overnight, but also through the winter. These roots will sit there, not much carbon moving around, so the fungi use them as a sort of overwintering survival structure. And they're also within the root, that environment is much more stable than the soil is, so it gives them a bit more protection. And these fungi do produce spores out in the soil, and that helps them reproduce. And they can also reproduce from these vesicles. So sort of hedging bets of having all this storage. And normally, I would say these are the rarest of the structures produced in the roots. Not always the case, but usually. And there's an odd one out. I think you can probably see which farm is causing the data to be everything else sort of squished down onto the axis here. So normally I would expect this data to be very low. We often see none at all. And in fact, in a few of these samples, there were none at all, which is quite ordinary. This one farm here, SC, almost all of the roots look like this, really chock full of structures. And actually, I probably should have had a play around with the axis of the graph so that it would demonstrate the differences down here a bit more clearly. But to fit this one on, I had to sort of scrunch it down a bit. But again, hopefully you can see lots of difference between samples. And I found it quite interesting. There was no clear difference really between the farms identifying as conventional or regenerative or organic. Previously, we had shown that there was very clear trend among them, less so here. And I think partly that's going to be due to weather factors, as Craig and Maria have mentioned, very dry year. And these structures in the roots, they're all very ephemeral. So they are very responsive to what's going on in the soil. So potentially that's what they're reacting to. But again, I would say very exciting. This is a lot of fungal structures generally. So even the ones here that look low, trust me, they're high. What we've got is some that are high and some that are very high. So that was all from me in terms of data. I would just like to say again, thank you all for sending in your samples. Fantastic to have that response. And a couple of take-home messages. Really high variability within and between the fields that were sampled. And as I say, no real trends that were immediately jumping out between the conventional and regenerative and organic farms. It will be exciting to see next year's samples where we've got sort of paired different crop varieties or different treatments to give us more clues about what's going on. So this year's data, you see it as a sort of extra baseline data. And then on top of that, we'll get extra treatments next year. And having said that, everything in these slides, everything I've showed you is very dead and we can't do any DNA work. Of course, Maria has done this metagenomics, so we can link together our samples and we can find identities for these fungi as well as just abundances. So that's all from me. As I think has been mentioned before, just drop questions in the Soil Circle or my email is there. I think we also have some time now for some discussion. So I'll hand back to Laura.
Thanks so much, Tom.
Thank you.
Great. So I don't know if anyone's got any questions, but if you'd just like to raise your hand if you do, and we can try and get as many in before the end of the session. So are there any questions from our farmers for our partners? I feel like that seems there's not many hands going up. So is that saying that you've learned everything you possibly can from the work that they've presented back to you today? Yes, well done, guys. Well, if you do have any other questions, that pop up after the session, we will be sending the recording around. After that, feel free to go onto the Soil Circle, ask questions. Our partners are involved in that chat, so they can come back to you on there. Just as a reminder, so we've got our reports that we'll be sending out mid-March. So that's the second week of March. The UK ETC are working on at the moment, pulling together all of that data. So if you've got any follow-up questions against those reports, take them to the Soil Circle. Just as well, we've got our Root Ranger packs that will be sent out by the end of this month. That will include all of the details around the protocols for this year. There's not a huge amount of changes from last year. One thing that we do want to flag with you is for your John Innes and Pez samples. That's the samples that are sent to Maria. Please, could you make sure that there's at least 500 grams per sample, just so that we've got enough soil for her to undertake her analysis. Dan, thank you very much, and thank you everyone. That's all I've got to cover. As I say, any questions that you think of, please add them to the soil circle, and the partners will be happy to come back to you. Great, and thank you to our partners for